Right, well, morning everyone. Now, given the theme of the conference today, I thought rather than give a more kind of results-driven presentation, I'd give a bit more of an overview to aspect of my PhD research and why it's important, why I do it. So as you can see, my research kind of focuses around vertebrates and skeletons. So I guess an important part to start off with is define what a skeleton is in the first place. So Merriam-Webster define it as a usually rigid, supportive or protective structure or framework of an organism, which is a very valid definition. But I think for purposes of my talk and what I'm going to be talking today, I use definition of it's any mineralized structure that can be produced by an organism. So that's something I want you to kind of keep in your minds during the presentation. So there's two major types of skeletons. The first one is what we call an exoskeleton, or basically means an outside skeleton, exo, outside. And this is the most dominant form of skeleton that we see in the natural world. It's expressed by most major animal groups. So for example, corals, starfish, mollusks such as snails, their shell would be considered as a skeleton, and arthropods, which are the most dominant group of organisms on the planet today. That's like creepy crawlies and stuff like crabs, lobsters, etc. So the other type of skeleton is called an endoskeleton, or an inside skeleton, and this is what is expressed in what we call vertebrates. So this is a very diverse group of probably the most characteristic organisms on the planet and throughout Earth history. So we've got stuff like fish, birds, reptiles, and ultimately mammals, which leads into humans ourselves. So an important question to answer is really, well, where and when did the vertebrate skeleton evolve and develop? And that's where my study organism comes in, which is the conodont. And these are really small, note scale there, elongate eel-like animals that evolved about 500 million years ago during what we call the Cambrian period, and disappeared around 200 million years ago at the start of what we would call the Jurassic period, or basically when all the dinosaurs started to evolve. Well, miraculously, it's still a pretty recent discovery. The animal was only really discovered in the early 80s, and that's because it's entirely soft-bodied, meaning that preservation is incredibly rare. But then I guess you're kind of wondering, well, if it's soft-bodied, where's the skeleton then? Which is a good question. So with conodonts, the skeleton is located in the mouth and is composed of these tooth-like elements that form complex apparatuses used for food processing. And these are composed of calcium phosphate, which is a very similar sort of material to what our own teeth are made out of. And this is generally what conodonts are known from in the fossil record. So I've got a really nice, hopefully this works here, it's a lovely little animation by Nick Gudemon, who's at the University of Lyon. Hopefully this works, there we go. Just to kind of give you a bit of an illustration about how that would have worked in life. So here we go, this is the apparatus here. So this is inside the mouth of the animal. So basically, it all works as a sort of grasping mechanism, and it brings the food to the back of the throat where it's then sort of processed. Just going to skip. There we go. Okay, so what I want you to know is that there's two major types of conodonts. First one is what we call a paraconodont, which is the earliest form of conodont from about 500 million years ago, and the elements are composed of a single tissue type. And the other type is what we call a euconodont, which is basically a more evolutionarily advanced form of conodont, and the elements are composed of two different tissue types. And the conodonts I focus on are the paraconodonts. So kind of more simple cone-shaped versions of these paraconodonts are really kind of well studied, well understood, and are seen to develop and grow through what we call basal accretion, which if you look at D to H, it's basically just the continual addition of tissue to the base of the structure until it is fully formed. And because of this knowledge, we know that paraconodonts must be directly ancestral to euconodonts, as the structure of a paraconodont element is identical to what we would call a basal body in a euconodont, which you can think as analogous to dentine. Euconodonts differ in that they have something called crown tissue, which is analogous to enamel. So you can sort of think of euconodonts as paraconodonts with like a little hat on or something. So this is all well and good. So what's the problem here? And that's where these things come in. Paraconodont elements called Westergaard-Adina, which, unlike the simple cone-shaped kind of forms, are a lot more structurally complex, both in the outside form and internal structure. And again, unlike cone-shaped forms, we don't really have any evidence of an evolutionary descendant of them going into the euconodonts. And the internal structure is very complex, and the growth and development of these elements is very poorly understood. So really, ultimately, these things, they just don't really make sense. So, and it really muddles our understanding of the evolution of the vertebrate skeleton. So what we really need to try and do is to try and resolve the growth and development of these problematic elements. So how are we going to do that? Well, I'll take you back to this figure that I showed you a few minutes ago. And this was created through advanced kind of 3D imaging techniques that allow us to look inside fossils and kind of reconstruct growth based on their internal structure. And this method is the most wonderfully named synchrotron radiation X-ray tomographic microscopy, which no one refers to it as that. We just call it synchrotron imaging. It's basically highly advanced kind of X-ray imaging. You can kind of think of it as giving a fossil like a intense CT scan or something. So to do this, we go to the Paul Scherrer Institute in Villingen in Switzerland, and particularly to this building here, which is called the Swiss Light Source, which I'll refer to as the SLS. So you can sort of think of the SLS as a giant donut with science inside it. It's this huge circular industrial building, and constantly running around it, we have this kind of ring where they can constantly fire particles, accelerate them around 24 hours a day. And at various points, I don't know how well you can see it on here, but they kind of deflect off into what we call beam lines. And each of these beam lines has a really kind of specific kind of property, which can be used for various different things. So the one that we use is this one up here, which is called the Tomcat beamline, which is probably the most forced acronym ever made. And what this is basically an intensely kind of focused laser of X-ray radiation that we can use on really small structures. So here's an example of how tiny these fossils that I work with are. And this is why we use this method. It works for very small things like this. So what we do is we kind of put it in this machine here in front of the beamline, and then it's zapped and scanned with this X-ray radiation. And what we get out of it is something like this, which is basically a really high resolution X-ray scan of the fossil. And again, note the scale here. This is about a 20th of a millimeter here. So this is the sort of size we're dealing with here. And as you can see, we get a really nice picture of the internal structure. So these here are just cracks from preservation of the fossil. But we also have these kind of dark lines here, which we would call growth lines. And you can think of those sort of as analogous to tree rings in that they record growth and development within the fossil. So from that, we can use some specialist software to do a bit of sort of, I guess you could call advanced coloring in order to segment the fossil into growth stages. So each of these colors is basically a package of growth within the structure. And in true Blue Peter fashion, here's one I prepared earlier. Unfortunately, it's not entirely complete, and I've got a lot more data to get through. Hopefully, I can get back doing that. The whole COVID situation has put a bit of a dampener and pushed that back a bit. But just this is an example of how we can help to show growth and development in these structures. And so the final thing I want to talk to you about is another method we can use, what we call small carbonaceous fossils, or SCFs. And these are essentially originally mineralized fossils that have had the mineral component removed during fossilization. So what we get left with are these organic films with the internal growth structure recorded in these dark brown lines that you can see. And this gives us a large data set to examine without the need for using advanced imaging techniques like those that I showed you before. Unfortunately, we only see them in one dimension or 2 dimensions. So we need to use this in conjunction with that synchrotron technique I showed you in order to help understand better how these things grew, and therefore how they sort of fit into the picture of skeleton evolution in vertebrates. So that's me pretty much done, but I just want to leave you with this element in particular and just ask the question, who wore it better? Because I think it's a pretty close call. And in fact, I polled the people of Instagram, and Bowie just edged it with 53% of the vote. So it would be interesting to see what the reaction from this group would be for this. But yeah, anyway, that's enough from me. Thanks for listening. Any questions?